Hello, and welcome to the webinar today entitled Targeted Degradation of the Enhancer Lysine Acetyltransferases, CBP, and p300. My name is Alexis Krause of LabRoots, and I'll be your moderator for today's event. I'd like to thank everyone for attending today, and I'm happy to welcome our featured speakers for today, Dr. Christopher Ott and Dr. Tim Geiger. This webinar is supported by Bio-techne. Bio-techne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tokris Bioscience, Protein Simple, Advanced Cell Diagnostics, and Exosome Diagnostics to provide the scientific research community with a comprehensive and world-class product portfolio of reagents, media, extracellular matrices, small molecules, assays, and instruments. Before moving into the talks by our featured speakers, I have just a few housekeeping items to address. Throughout the webinar, we invite you to submit any questions you have for our presenters using the Ask a Question box located on the left side of your screen. These questions will be asked to the presenters directly following the presentation. In addition, right below the Ask a Question box, you will see a tab called Handouts. In that tab, you'll find extra content that relates to today's webinar. And now I would like to introduce our speakers for today, Dr. Christopher Ott and Dr. Tim Geiger. Dr. Tim Geiger is a molecular and cellular biologist who has enjoyed a varied career from his postdoctoral work developing in vitro transcriptional recruitment models to overseeing research teams in diagnostics and pharmaceutical analytical development to business development and high-throughput immune sequencing. He also guest lectures at UC San Diego on omics and translational medicine. He is an early adopter from the Simple Western technology before joining Bio-techne, where as a field applications manager, he continues to focus on the Simple Western product line. Dr. Christopher Ott is a chemical biologist at the Center for Cancer Research at Massachusetts General Hospital and Harvard Medical School. The principal focus of his research is to determine mechanisms of malfunctioning gene regulation in cancer and to develop therapeutic strategies to modulate gene regulatory factors in tumors. His team is developing early-stage chemical probes and prototype drugs directed at modulating critical gene regulatory factors, including the first chemical degraders of the enhancer acetyltransferases, CBP, and p300. I will now turn it over to Dr. Geiger for the presentation for today. Welcome, sir.
All right, well, thank you for that introduction. I'm going to give you a quick introduction to Simple Western before we get into Chris's presentation. Okay, so what exactly is Simple Western? If you're joining this webinar, you probably already have a sense on what Simple Western is. Fundamentally, we're a replacement for Western blot, right? But Simple Western is really more akin to a capillary electrophoresis instrument for protein separation upstream of detection by an immunoassay, specifically an indirect immunoassay or an indirect ELISA. And we are, as a company, are dominant players in capillary electrophoresis with our instruments supporting biologics development and testing throughout the biopharmaceutical industry. So we sell the ICE family of instruments, with our flagship instrument being this one here, the Maurice, which on the left here, which does both size and charge-based separation for analytical characterization of biopharmaceuticals. And this is data that is well accepted by the FDA and other regulatory agencies in this space. Bio-techne is also a leader in immunoassays. We have a large catalog of proteins and antibodies through our Novus and R&D Systems brands. Anybody in the ELISA space knows our quantikine kits. They're considered gold standards in the validated ELISA space. And we sell Luminex reagents, and then we have this Ella platform, which is an automated benchtop ELISA. So it's really these two technologies that merge to create Simple Western. Ironically, considering the name Simple Western, the one thing that we are fundamentally not is a Western blot, and I will explain that. So we're next generation for that technology, but we're definitely not a Western blot. I'll explain that in the next slide. Okay. So I'm going to show you a video. This is of the Jess, but the Wes is fundamentally the same look and feel. So there's fundamentally two big leaps forward with Simple Western, and that is relative to a Western blot, you get much faster results, and you get quantitative reproducible data. An experiment on this is fairly easy to set up once you're familiar with it. There's two things that you're loading into the system. One is this capillary cartridge that you see on the top here. This is a 25 cap cartridge. You also have a 13. So you have enough room here for 24 samples plus your molecular weight ladder. And then we have this plate. So you're just going to load your samples and then all your immunoassay reagents, so basically your antibodies and solutions, into the plate. It's already preloaded with separation reagents. And then once you've loaded the plate, set up the plate, then you just load it into the instrument plus the cartridges, close the door, and hit start. The system is going to start by preparing the capillaries for the protein separation phase. And as I mentioned before, the proteins are separated akin to size-based capillary electrophoresis, or CESCS. And like that system, Simple Western uses a linear polymer separation matrix. So we're also using a discontinuous separation setup with a stacking matrix here. All right, so the next step then is sample loading. And I mentioned that Simple Western has a lot lower, it's a much more precise instrument, less variation than you would deal with a classic Western blot. So there's a few different things in classic Western blot that kind of encroach error into the assay. And pipetting error is definitely one of those issues because you're loading your sample directly into the gel. We minimize that source of error because your sample is loaded into the well in excess, and then the instrument controls how much of that sample actually gets loaded into the capillary. And it's pretty little. It's less than 100 nanoliters of sample that actually gets loaded in. All right, so now your capillaries are all loaded. Now they're going to move over into the running buffer. Voltage will be applied, and the proteins are going to be separated by size. Okay, so this step right here, this is why we are not a Western blot. By definition, a Western blot involves the blotting of proteins onto a membrane. And that is, again, a major source of variation in the classic Western blot. So we've eliminated that step by photo cross-linking our proteins onto the capillary surface. Now we can do the entire assay within that capillary. So now that your proteins are photo cross-linked, the system can flush out all the separation reagents, come in with a blocking buffer, primary antibody, which is going to come through in a sec. There's your primary antibody, and then that'll be followed up by a secondary antibody and detection. All right, with the Wes, you're going to use chemoluminescence for detection. With the Jess, you have chemoluminescence, but you also have two fluorescence channels. You have a protein normalization channel for total protein normalization. So basically, you have 4 channels of detection with the Jess. And then we also have this new reagent, the replex reagent, that you've hopefully heard about. And this can remove the antibodies from the first probing so that you can come in and do a second probing on the same capillary. So you're increasing your multiplexing even further with that. So when you consider that each capillary is independent and loaded from separate wells, you can make highly complex experiments by doing different reagents in different capillaries. So in the end, you can make a pretty highly complex, a highly modular, highly multiplexed assay if you're trying to address very complex scientific questions. But remember, you don't have to do that, right? If your current Western blots are working for you, the system still is advantageous. I mean, if you're just looking to accelerate your science, this system gives you results in as little as 3 hours. Okay, so here is the whole Simple Western family of products. I've already introduced the Wes and Jess. Again, these both load up to 25 cap cartridges, so you can again load about 24 samples plus your molecular weight ladder. And then we also have these 3 large format systems. I'm just showing 2 here, but these all load up to 88 of your samples And it's run all at once, but it's done over 8 cycles. And these are our SaliSu, our NanoPro 1000, and then our PeggySu instruments. And the SaliSu does size separation, NanoPro does charge, and PeggySu does charge or size-based separation. It does both. And again, in terms of advantages, the system is very fast, results in as little as 3 hours, and it's fully automated, right? So you can go off and do other science while you're waiting for this. It's nimble. You can have very simple experiments if that's what you're looking for, but you can do very complex experiments as well on it. And then something I haven't highlighted before is the low sample volume. Again, I consider this something that accelerates your science because you spend less time producing enough sample necessary to be able to do larger experiments because you can work with these small volumes. But again, speed is nothing if you cannot generate good quality data. And our automation gives you the precise, reproducible data you're looking for. And then our software, which is really user-friendly, makes it easy to report out quantitative data with our system. And the title of the slide, Science is Only as Precise as Its Weakest Link. This is why we at ProteinSimple like working closely with all of our Bio-techne sister divisions and brands. Bio-techne produces a lot of different reagents and scientific solutions to be able to perform science, and then provides animal tools and then to generate accurate and meaningful data with reagents. And this is one nice example. This is work we did, working very closely with our R&D systems group to develop the SARS-CoV-2 serological module. And I'm not going to go into details about this module, but I'm just using it really to highlight the capabilities of the system with a well-developed assay. So what you're looking at in this left graph, this is an electropherogram. We're doing a titration series of these samples, and you can see this is our SARS-CoV-2 ladder. And if you can't see the labels, it's RB2, nuke, the S2 and S1 subunits, and then full-length spike protein. So on the x-axis is molecular weight, and then on the right, on the y-axis is chemoluminescence. And this was a dilution series done in duplicate. So each one of these traces here, you can see it's paired up. And what's striking about it is if you look at the graph on the right now, this is that same data reported out. And in this case, you've got chemoluminescence, which is area under the curve on the left, on the y-axis, and then dilution on the x-axis. And remember, these are duplicates. You can barely discern the fact that there are 2 points at each one of these titration points on these curves for these 5 proteins. Really, the only one where I'd say you start to see the points significantly diverge is this lowest data point here on the far right. So I'd probably argue that that last data point is falling out of the quantitative range of the assay. But certainly, excluding that single data point, you're looking at, at least 3 logs of linearity in this experiment here. It's really, really nice data. Similarly, we've been working closely with our Tocris brand, and they generate small molecules and peptides to aid in life science research. One of the platforms they've licensed is the PROTACs, and these are going to be highlighted in Dr. Ott's talk today. These are bifunctional small molecules that recruit the E3 ligase to your target of interest through this linker region here. And by recruiting that E3 ligase, this leads to polyubiquitination of your target and then subsequent degradation through the ubiquitin degradation pathway. Just a general statement, it's hard to make breakthrough biological discoveries if you're limited by the precision of the tools you're using to study those biological changes. So again, this is why Simple Western is a really good complement to things like these PROTACs. All right, one last slide. I just wanted to quickly call out a milestone that we at ProteinSimple are really proud of. Simple Western has just crossed the 1,000 publication threshold. And we're well represented, as you can see on the right here, on some really high impact journals. So it's just really great to see how well adopted Simple Western has become over the last few years. All right, well, thank you then, and I'm gonna go ahead and pass it on to Chris.
Thanks, Tim, for the opportunity to present some recent work on behalf of my research group, which is specifically focused on a tool compound that we've recently described, one of these PROTAC molecules that targets two important acetyltransferases, CBP and p300. This work comes out of my laboratory for chemical epigenomics, which is based within the Center for Cancer Research at Massachusetts General Hospital and Harvard Medical School, and you can read more about our work and interests at our website, otlab.org. So any discussion of our approaches to chemical epigenomics as it relates to cancer biology needs to begin by framing how we think of the epigenome and its impact on the hallmarks of cancer cells. Conrad Waddington first proposed the conceptual framework of the epigenome, the deterministic cellular signaling that occurs on top of our genome, as a series of hills and valleys undergirded by genetic guy wires that enforce a particular cell state as a cell becomes more differentiated. And in cancer, genetic mutations disrupt this guy wire scaffold, in turn leading to alterations in the height and depth of these peaks and valleys, which affords a differentiated cell the potential or the ability to sample alternative cell states that can eventually facilitate the outgrowth of a malignant clone characterized by hyperproliferation and resistance to apoptotic cell death, the hallmarks of cancer. And while there are many signaling platforms that make up the epigenome, the most proximal to genes themselves is chromatin, the nuclear structures that fold and organize the genome in both highly compact, heterochromatic, or more loose or accessible euchromatic structures. And there are a huge number of chemical features that make up chromatin, including post-translational marks of histones, transcription factors that bind directly to DNA, the chromatin-associated factors that can both modify nucleosomes, modify transcription factors with post-translational marks, can move nucleosomes out of the way of specific regulatory elements of the genome. Of principal interest to my group are enhancer elements, shown up here in the left, which are emblematic of active chromatin and mediate the transcriptional activation of genes that have restricted or more cell type specific expression. Enhancer activity is primarily mediated through the direct DNA binding activity of transcription factors. And we think of transcription factors as sort of the integrators of genetic to epigenetic signals. They bind to DNA in a DNA-specific manner through their DNA binding domain, and so they require a specific genetic input for their activity, and then they signal to other functional domains, to components of the epigenome, to induce macromolecular formation, ultimately chromosomal looping, recruitment of RNA polymerases to activate gene expression and polymerase elongation, and ultimately RNA production. There are a lot of features of enhancers, characteristics of enhancers. They can be very distal from the promoters that they regulate. They're hypersensitive to nucleases. They contain very specific histone modifications, including lysine methylation and lysine acetylation. The first descriptions of enhancer activity by Walter Schaffner and his group way back in the early 80s revealed two distinct classes of enhancers. Enhancers like the viral SV40 enhancer that seem to be ubiquitous and active across many different cell types. And then enhancers like the immunoglobulin enhancer, which had very specific restricted cell type activity here, specifically lighting up a reporter gene specifically in myeloma cells and not in HeLa cells. And larger efforts, more modern efforts like the Roadmap Epigenomic Consortium and the ENCODE Consortium have basically proven this principle, and that there are classes of enhancers that are ubiquitous across tissues, and that there is a subset of enhancers across tissues that is very cell type specific. And these are the principal mediators of cell type specific gene expression. Presciently, in their 1983 Cell paper, Schaffner and his colleagues supposed that aberrant enhancer activity could actually lead to diseases such as cancer. And recent research has dramatically expanded our understanding into really how significantly enhancers can contribute to cancer, many different types of cancer. And so my group at MGH is focused on developing tools and insights to test the hypothesis that direct targeting of enhancers may be a rational approach for cancer therapy. This involves developing screening assays, new ligands, and new chemistry to target the transcription and chromatin factors that establish and maintain enhancers, and also utilizing high-resolution genome-wide profiling technologies to discover response and resistant biomarkers to this new chemistry, and mechanistic insights into the activity of synthetic enhancer modulators, whether they be small molecules or slightly larger molecules, such as the one I'll describe in this presentation. And one aspect to enhancers, or a characteristic of enhancers that we are particularly focused on, is their high level of lysine acetylation. Lysine side chains throughout enhancer structures can be highly acetylated, both on histones, transcription factors, other chromatin regulators. And they're acetylated through the activity of HAT or CAT, lysine acetyltransferase enzymes. And this acetylation is highly dynamic. So while the CAT enzymes are the writers of this acetyl mark, HDACs, or histone deacetylases, remove these post-translational marks, or the erasers. And integral to affecting this chemical signaling are proteins that contain a specialized protein module called a bromodomain, which is a specific reader of lysine acetylation. Proteins with bromodomains read combinations of acetylysines at enhancers, which affect signaling at enhancers through either subsequent protein-protein interactions or enzymatic activity. Well, bromodomains are these specialized acetylysine binding modules. There are 46 proteins in the human genome that contain a bromodomain. Some contain more than one bromodomain. Their structure is very unique. They're a left-handed bundle of 4 alpha helices, and they mediate binding to acetylysine through a conserved asparagine that is in the middle of the pocket there. And while this asparagine-mediated recognition of acetylysine is highly conserved, it's the flanking residues of bromodomains that are actually quite distinct from one another and confer specificity to binding specific acetylated lysines on different epitopes. One of the bromodomain factors is probably most well studied, and one that I was very interested in during my postdoctoral studies was this protein, bromodomain-containing protein called BRD4, which is very highly active in enhancer elements. It's an epigenetic bookmark, if you will, of active genes remaining on chromatin through mitosis, and functions by recruiting the p300 complex to active promoters and able to activate elongation of RNA polymerase II. During my postdoc over at the Dana-Farber Cancer Institute in Jay Bradner's lab, there was a chemist in our group, Jun Chi, who developed the first synthetic small molecule ligand that specifically bound to the bromodomains of BRD4 and closely related members of this bromodomain family called the BET family, or the bromodomain and extraterminal family of proteins. And this compound has remarkable activity, and lots of people have been studying this molecule in lots of different facets of chromatin biology and cancer biology. And one piece of data that I was particularly fascinated with was this piece of data generated in our lab and in collaboration with Konstantin Mitsiadis' lab, where they found that treatment with JQ1 of a myeloma cell line led to potent cell death and specific depletion of BRD4, specifically on the immunoglobulin enhancer. That same enhancer that Walter Schaffner first described as being the first cellular enhancer and specific to the B lymphoid lineage. In this cell line, this enhancer is actually oncogenic. It's translocated to the MYC locus, so it actually forces high expression of the MYC oncogene. And through displacement of BRD4 with this small molecule from chromatin, you release BRD4 from this oncogenic enhancer, leading to downregulation of the MYC oncogene. And so I followed up on this work and looking in other contexts of B lymphoid malignancies with David Weinstock and his group studying B-cell acute lymphoblastic leukemia, where JQ1, this bromodomain binding ligand, is very highly active, and also in chronic lymphocytic leukemia with Jennifer Brown and her lab, where the molecule is quite active, both in vitro models and in vivo models of the disease. But one aspect of bromodomain ligands is that they just target the bromodomain. And in some cases, this can lead to very potent displacement of the protein from the acetylysins that they recognize. But a lot of proteins that contain bromodomains actually have a lot of other functional domains, which are not actually inactivated with bromodomain ligands. And so two of my friends who were co-postdocs with me in Jay Bradner's lab over at the Farber came up with this idea where they could potentially link JQ1 to a ligand that binds to an E3 ligase, in this case thalidomide, which binds to the E3 ligase cereblon, which then would potentially hijack cereblon to induce protein degradation through the ubiquitination of in this case, BRD4. And this worked remarkably well. They described ternary complex formation with this molecule, this linked molecule, called DBET1, with this rather modest linkage, and very quickly could observe degradation of and loss of BRD4 protein with treatment of this molecule. And so this concept actually extends this concept that was really pioneered by Craig Cruz and Ray Deshays, this idea of proteolysis targeting chimeras, in which you have a synthetic molecule that basically brings in the close proximity of protein of interest and an E3 ubiquitin ligase complex in order to selectively degrade through the ubiquitin proteasome system of protein of interest. And so there's been a lot of work in the past few years describing the system and different compounds that can degrade different proteins of interest and hijack different E3 ligases, hijacking different E3 ligases depending on the ligand used. And so when I started my lab a few years ago here at MGH, we decided to try this concept to degrade several proteins that had not been degraded before by this mechanism. In particular, these two lysine acetyltransferases, p300 and CBP, for which a lot had been studied and a lot of ligand development had been done, but had not really been effectively degraded by this type of chemistry before. And we were particularly interested in these proteins because they have they're chromatin-associated proteins and enhancer-associated proteins that have a lot of different functional domains to them. So they're part of a unique class of proteins that have both a acetylysine reader domain through the bromodomain, but also a lysine acetyltransferase domain. And a lot of different aspects of the biology of these proteins has been studied. They're very highly homologous, so at the domain level, they're typically over 90% conserved. They dynamically acetylate over 21,000 lysines in the proteome on over 5,000 different protein substrates. And this exemplified in Chuna Chowdhury's excellent paper in Cell just a few years ago, where they really profiled the dynamic acetylome of CBP and p300. These factors are required for enhancer-mediated transcription, and there has been a lot of ligand development for the different domains of this protein, of these proteins, in particular the bromodomain and the lysine acetyltransferase domain. And these molecules have been developed to be quite potent, but they have differing effects on the protein, and neither inhibitors of the cat domain or the bromodomain can completely ablate the activity of the protein. Interestingly, when you combine these molecules in cell culture and treat cells with molecules that target the bromodomain and target the acetyltransferase domain, they can have quite potent synergistic activity in cancer cell line models. Here, described by Phil Cole and his group in a paper a couple of years ago in a prostate cancer cell line. And it seems like when you inhibit both the cat domain and the bromodomain, you have a sort of an accelerated or a highly effectual displacement of these proteins from chromatin, probably mediating this synergistic effect. So we thought, well, what if we made a PROTAC version of these molecules to induce proteasomal degradation of CBP and p300, and how would that compare to inhibition of the bromodomain itself, or the cat domain itself, or the two inhibitors combined? And so we were lucky in that we have a lot of choices in order to design a potential PROTAC degrader of these proteins. We could choose to use either a bromodomain ligand, of varying potency that have been developed by a number of different groups. Here, the most potent selective being this compound GNE781 that was developed by Genentech. Or we could use a haptomain ligand or a cathomane ligand. The most potent, at least the time when we started this work, was developed by AbbVie Pharmaceuticals, this molecule called A485, which is quite potent and a quite potent inhibitor of the acetyltransferase activity of these proteins. And so while we had a lot of choices, we didn't have a lot of bandwidth with our chemistry. So we needed to make a decision as to which molecule we were actually going to make a PROTAC version of. And beyond just what compound we would choose to link a thalidomide-based cereblon degrader to, where would we actually attach it on the molecule? And here is where we did computational modeling of the ternary complex of the E3 ligase cereblon bound actually to the bromodomain or the acetyltransferase domain of these proteins. And what we did was we basically did use this program Rosetta docking, which and docked either the bromodomain or the cathomane thousands and thousands of times against cereblon and looked for minimal energy poses and just basically measured the distance between the different atoms where we could potentially attach a linker and the thalidomide ligand bound to cereblon. And what we found was, and when we just basically graphed the distribution of the distance between each of these atoms and thalidomide, what we found was, first, that the bromodomain seemed to give us the most minimal energy poses when compared to the acetyltransferase domain. And when we looked at different potential exit vectors for linker attachment within this GNE781 molecule, it seemed as if this pyran ring system would basically give us the most sort of minimal energy poses when docked into cereblon. And so that is the molecule that we ended up making. Here's some examples of some of these poses of the bromodomain. Here, the CBP bromodomain docked into cereblon. You can see that these poses specifically all have potential linker distances under 20 angstroms. And so we designed this molecule. DCBP1, which has a PEG-4 linkage between here thalidomide, which binds to cereblon, and GNE781, bound to what was the pyran ring system. And this PEG-4 linker has an approximate distance of about 20 angstroms, which in theory would allow for ternary complex formation of all these different poses that we predicted here with this linkage system, if you will. And so to describe this molecule in activity assays, we turn to the protein simple Wes, as Tim highlighted. This was a machine that I procured basically as soon as I started my lab because I knew we would be doing a lot of immunoblotting and immunoassays, and we wanted to be able to profile the activity of these compounds, these classical compounds, very quickly. And so all of the blots that I'll show you in these subsequent slides are actually generated by the protein simple Wes. So they're not technically Western blots, but they're capillary immunoassays. Which actually worked quite well for CBP and p300. These are very large proteins, they're 300 kD. And so one of the real highlights and real enabling features of using the protein simple system, the Wes system for these assays, they allowed us to profile the activity of DCBP1 very rapidly. So in 3 hours, we can go from lysate to figuring out if this molecule worked or not. And the molecule works quite potently. So you can see here's a dose response activity of DCBP1. Degrading CBP at concentrations less than 10 nanomolar. You can see within 2 hours of treatment with the compound, we get almost complete degradation of both CBP and p300 with DCBP1. This activity can be rescued with saturating doses of both free ligand on the CBP and p300 side, the GNE molecule, and pomalidomide on the as a E3 ligase cereblon ligand. This activity can also be rescued by blocking the proteasome or netylation of the E3 complex itself. This activity is also cereblon specific, so in a cereblon knockout cell line, we see loss of the activity of DCBP1. We also constructed a molecule which we call DCBP1 bump, which actually abrogates its binding to cereblon itself by adding a methyl group on the right side of the molecule here. And you can see that DCBP1 bump has no degradation activity at all. Here. We also have very selective effect of CBP and P300 over BRD2, BRD3, and BRD4, other bromodomain-containing proteins. So we do not see, while a degrader of these BET proteins is quite active, here DBET6, our DCBP1 compound does not degrade any of these proteins. So then we wanted to address, well, we're a cancer biology lab and a cancer epigenomics lab, and so what cancers may be most susceptible to a degrader of CBP and P300? And so we turned to a CRISPR knockout data set developed by the Broad Institute and the DETMAP group over there, where they've done genome-wide CRISPR knockout screening across hundreds of cancer cell lines. And we just asked the question, well, what are the cell lines and the tumor types most sensitive to CBP and P300 knockout? And unambiguously, these were multiple myeloma cells. You can see here the dependency of multiple myeloma cell lines. Here, about 20 different cell lines that are dependent on CBP, and here even more so on P300. And so we thought this would be the best sort of first shot on goal in sort of profiling DCBP1 and its activity in a cancer model system. This is the cell line MM1S, which I mentioned before was hypersensitive to the BRD4 ligands. You can see we also have complete degradation of CBP and P300 at doses less than 10 nanomolar here, and within 2 hours of treatment of DCBP1. We see this observation across multiple different multiple myeloma cell lines, MM1R, KMS12BM, KMS34. This molecule in growth viability assays is quite potent and significantly more potent than the inhibitors of CBP and P300, either the acetyltransferase inhibitors or the bromodomain inhibitors. And we see that this activity is sort of accentuated both in sort of an IC50 shift, but also in a shift at Emax, which is likely indicative of this molecule inducing apoptosis in these cell lines. In growth over time assays, we see that DCBP1, this is 100 nanomolar treatment, can potently reduce cell numbers in vitro within 48 hours of treatment, whereas equimolar doses of the inhibitors or even combination of the inhibitors have a rather cytostatic effect at the same dose. By profiling the transcriptome to try and understand how this molecule is working, we can see that DCBP1 treatment has a very distinct effect on the multiple myeloma transcriptome. We find that the gene that is most downregulated, because this is a cell line where the potent immunoglobulin enhancer is translocated to the MYC oncogene, the MYC gene is the most downregulated gene of the entire MM1S transcriptome with DCBP1 treatment. And this MYC downregulation at the transcript level is highly attenuated, if you will, when compared to equimolar doses of either the CAT domain inhibitor or the bromodomain inhibitor, or combinations of the HAT and bromodomain inhibitor. You can see at the protein level, MYC is potently downregulated within 6 hours of treatment. We look at gene response signatures by gene set enrichment analysis, and we can see that the MYC signature is the most affected gene signature at the transcript level with DCBP1 treatment. When we look at the proteome level, after 3 hours of treatment, DCBP1, we can see specific downregulation of the target, CBP and P300, and also the transcriptional target, MYC. We don't see significant downregulation at the protein level, again, of BRD4, ALOS, which is the target of pomalidomide and lenalidomide, binding to cereblon in multiple myeloma cells, or also the known off-target cereblon modulator, GSPT1. However, at 6 hours, you can see that a vast amount of the proteome is affected with DCBP1 treatment. So I would encourage you, if you use DCBP1 or other CBP, p300 degraders in your studies to focus on early time points if you really want to tease out the specific and acute effects of CBP and p300 degradation. At the epigenome level, we looked at enhancer acetylation with K27 acetylation chip, and we can see with degradation of CBP and p300, we have complete loss of lysine acetylation at enhancers of p300 and CBP binding sites. Shown here is that immunoglobulin locus that drives MYC expression, and you can see here loss of acetylation with DCBP1 treatment, where it's really only modestly attenuated with inhibitor treatment. And at the chromatin accessibility level with the TAC-seq, we see that enhancers with the inhibitors are largely at least remain accessible. Whereas with the degrader, we actually see loss of chromatin accessibility, which we think is loss of protein-protein interaction contexts that are mediated by CBP and p300, leading to loss of transcription factor engagement with the enhancer itself. And so we think that DCBP1 will be a useful tool for understanding sort of the effects of CBP and p300 activity at enhancers, how they nucleate enhancer activity, and the acute effects of their depletion on chromatin. And we think it would be a good tool for teasing this out in cell culture and in vitro systems. We also think that there's a potential for DCBP and DCBP1 or analogs thereof or other degraders of CBP and p300 potentially as therapeutics for cancer. You can imagine that complete ablation of enhancers with drugs may not be therapeutically tolerable. And so that's an area that we're beginning to explore, and we've done some here's some unpublished work, some recent work from the group, where we've actually been able to establish some in vivo activity of DCBP1. With daily dosing of DCBP1 at rather low concentrations, we can see by IP injection that we actually don't have much effect on the body mass of mice that are injected with DCBP1. So it seems to be well tolerated, and these are doses that seem to be active. So here we've harvested 4 hours after the second dose of DCBP1. We've harvested livers, and we can show, using the capillary system, the immunoassay, using the West system, we can show depletion, almost complete depletion in the liver of CBP and p300. And yet this seems to be well tolerated. So we think that this tool compound can also have in vivo utility for studying CBP and p300 depletion effects in vivo in mouse models. So with that, I'll stop. I'd like to particularly shout out my group, both current and past members, in particular Raghu Vanam, who led all of the chemistry efforts for developing DCBP1 in my group. And also, I'd also like to shout out Sam Ojeda in my group, who led all of the assays and did all of the assays on the West system to profile DCBP1 treatment in the various cellular model systems. I'd also like to particularly shout out John Sigilian in Mike Lawrence's lab, who did all of the computational modeling, and the folks in Willie Haas's lab here at MGH, who did all the proteomics analysis of DCBP1 activity. And I thank, of course, the support for our lab from various funding sources. And I'm excited to take your questions. Thank you very much.
Thank you, Dr. Ott and Dr. Geiger, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Looks like we've got some great questions coming in from the audience already. So let's start with this one. And Dr. Ott, this is for you. Is there a reason you decided to attach the linker to the 5 position of the thalidomide as the 4 position is more typically seen in PROTACs?
So when we first did our modeling studies of the potential ternary complex formation, there was some evidence that the 5-position carbon was slightly closer. And so we made the decision to do the linkage off of that position. Now, I imagine that a 4-position linked compound would work quite as well, would also work. So we don't have comparative data between the 2 linkages, but that was the reason we made that decision.
Great, thank you so much. So moving on to our next question. So current PROTAC development largely depends on empiric optimization of the linker length. Did you only synthesize the molecule predicted by Rosetta to fit best, or did you try several linker lengths? Yeah, so we made the decision up front actually to use a PEG-4 linker length. There's been a lot of PROTAC-based work using a lot of different linker lengths and linker compositions, and those are studies that we're pursuing now and seeing how those can affect the activity of the molecules. But we made the decision to make a PEG-4 compound because we thought that would kind of hit a sweet spot in terms of potential for degradation capability. And then we made the decision based on a PEG-4 linked molecule, which, based on the ternary complex modeling, which sort of exit vector to attach the molecule to in the GNA scaffold. Wonderful, thank you so much. We've got a couple more questions. So now, here's a good one. How do you process the samples before probing for EP300 on the West? Right. So in our assays, we use a typical RIPA buffer, and then we followed by some incubation on ice, and then we pass that lysate through a fine gauge needle in order to really do our best to comprehensively disrupt the chromatin structures within the lysate. We have also tried sonication of the lysates. We found that sonicating the samples led to a little bit more variable results. So we've been typically using a needle to pass the lysate. Great, thank you so much for that clarification. So here's another question. So how clean are the digital westerns for CBP p300? Is there only a single peak with used antibodies, or do you see multiple peaks at different molecular weights? Yeah, we did a fair amount of optimization on the West with a variety of different antibodies. We found that these 2 antibodies from cell signaling did work the best in terms of consistency of signal. In terms of how clean they are, in terms of the other bands and the number of bands that come up, this really depends on the cell type. So some cell types will show multiple bands or sort of a broad, really broad range in the 300 kD range, and some are quite clean in terms of a single band. Wonderful, thank you so much. We want to remind our audience, any questions that we're unable to answer today and those that come in during the on-demand period will be answered by our speakers via the email address that you provided at the time of registration. So please keep those questions coming in. We're happy to answer them all. So Dr. Geiger, we haven't forgotten about you. I have a question for you. So sure. How does the sensitivity of Simple Western compare with the Western blot?
Yeah, that's a good question. It's like with everything with an immunoassay, it kind of depends on the performance of the antibody. But we've done quite a few studies on a lot of different targets to try to understand the sensitivity. The average seems to be around about a 4-fold improvement relative to a typical Western blot. But again, it depends on the target, and there are some targets where we see significantly better sensitivity with Simple Western compared to Western blot. And that's reflected in some of the papers that have been published. I can think of one because it's someone I worked with directly as an FAS. A group out of Cal State Fullerton that published a paper on muscle fibers where they were able to get single fiber resolution of their targets, which had been a struggle to achieve with Western blot. But because of working with the West system, they were able to do that. And in doing so, they were able to really tease apart specific muscle fiber related signaling that otherwise has been convoluted by the fact that you always had to aggregate fibers to be able to do those studies. So that sensitivity can make a pretty big difference in your work for sure.
Great, thank you so much. So, Dr. Ott, we're going to move. Excuse me. We're going to move back over to you. How does the activity of DCBP1 compare to other degraders of bromodomain proteins like DBETs?
Yeah, so you know, we spent a lot of time studying BRD4 and the effects of BRD4 inhibitors and degraders on gene expression and chromatin structures. We haven't done much work comparing, you know, the DCBP1 compound with the DBET compounds. We can imagine that, you know, the activity of BRD4 at enhancers can be, you know, quite similar to the activity of CBP and p300, but also likely very distinct. BRD4 has very specific relationships to recruitment of p300 and activating elongation of the RNA polymerase complex. And so we can imagine that, you know, complete degradation of BRD4 may have very specific or distinct effects than a degrader of CBP and p300, and may lead to opportunities potentially for, you know, synergistic activity on enhancers and promoters together. But we have not, as of yet, done comprehensive head-to-head analysis of the two compound series.
Great, thank you. So we do have time for a couple more questions. So Dr. Geiger, we'll move back over to you. What data do you typically see people report out? Electropherograms or that Western blot style view?
Yeah, I guess it kind of depends on personal preference. I think what I've seen mostly is when people are internally analyzing their data, they quickly become acquainted with that electropherogram view. It's truly sort of the quantitative way to look at your data, and so as you get more familiar with it, you'll find that you really see a value in that way of reporting out the data in terms of just the power to be able to quantitate it, visualize it, understand any kind of like variability and stuff like that. The disadvantage of it, of course, is just that there's it's difficult to put a lot of data in a single small space. The classic Western blot, quote unquote, view gives you that power. And that's the reason, like, when I was reporting my data out and talking about quantitation, I was showing you guys electropherograms. But then when Dr. Ott was showing his data where he has a large number of samples that he wants to show in a single slide, it's advantageous to throw up that blot view. So a lot of times with papers, we do see that people use the blot view just to be able to encapsulate the information in a small space. But when you're looking at your own data, you'll find very quickly that you're going to really prefer and appreciate that electropherogram view.
Great, thank you so much. Back over to you, Dr. Ott. Has your work with DCPB1 or analogs shown any selective degradation for CBP or p300 over the other?
Yeah, that's a great question. So yeah, obviously it would be fantastic to be able to develop even more selective tools for one versus the other. And the answer is yes. We do see certain contexts and some analogs that actually show selectivity of one versus the other. We're trying to understand the molecular recognition events that mediate that selectivity. And so we're actively working on that now.
Wonderful, thank you. And how about, did they alter the heat denaturing step for the West samples?
I believe that's probably a question for us. No, we did not alter any denaturing steps. No.
Great, thank you so much. Okay, looks like we've got time for maybe one or two more questions. So, Dr. Ott, what is the optimal dose and time point I should use DCBP1 in my cell system in order to study direct effects of p300 CBP loss?
Yeah, another good question. So I think typically in most cell systems we see almost complete, at least by a Wes analysis, complete degradation of both CBP and p300 within 2 to 3 hours of treatment at about 100 nanomolar. So I think that's a good dose and time point to start. But with any cell system, you'll want to tune it based on your cell type. We have noticed some cell types where uptake of the molecule is slightly attenuated, so the dynamics take a little bit longer in order to cause degradation. But you'll want to be careful in terms of doing analysis at longer time points because, we've noticed by proteomics analysis that in some cell lines, at 6 hours, you see massive effects on the proteome beyond just CBP and p300. So if you're looking to measure, sort of acute primary effects of the loss of CBP and p300, you'll want to keep the time points short and sort of tune it based on your cell system.
Wonderful. Thank you so much. So here's another question. So for the immunoblot of CBP and p300, did you do multiplexing with Ventolin and GAPDH? And which one do you recommend for multiplexing with CBP p300 and Simple Western?
Yeah, so we typically multiplex with Vinculin because we use the high molecular weight kit, and Vinculin has a higher molecular weight that we could assay together with CBP and p300 together. So yeah, we just mixed the antibodies together and ran them in a multiplex fashion.
Great, thank you so much. Okay, so it looks like we've got time for maybe one more question. So, you know what, Dr. Ott, we'll go ahead and end with you. So for your modeling efforts to design DCBP1, did you incorporate conformations of various linker structures?
We didn't. We actually tried to keep it simple. We know that, there have been other groups that have used the Rosetta modeling package and different types of protein ternary complex modeling to actually model linker conformations to really try and anticipate what linker design may be optimal. We didn't do that. We're trying a little bit of that now, but we simply just made measurements between the two ligands on the respective proteins, and then sort of analyze the distribution of those of the direct lengths between the two ligands. We can imagine that in some cases that might not work perfectly. There might be steric issues with residues blocking that proposed linker or that proposed avenue, if you will. But in this case, by manual inspection, we didn't see any docking that would predict that. So we kept it simple and just went with the straight distance between the two.
Great, thank you so much. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information that you provided at the time of registration. We would like to once again thank Dr. Ott and Dr. Geiger for their time today and their important research. We would also like to thank LabRoots and our sponsor, Bio-techne, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay to share with your friends and colleagues. That's all for now. Thank you for joining us and have a great rest of your day, everyone. Bye-bye.